I'd like to first start by asking you all to take a second to think of a threatened marine habitat. Many of you probably thought of coral reefs, mangrove forests, or seagrass beds, but how many of you thought of oyster reefs? Oyster reefs, which grow in three-dimensional structures that support diverse communities of organisms, are among the most endangered habitats in the world, with an estimated 85% global loss, which is illustrated here in this map. We can see that the areas in blue, or good conditions, are only found in two small regions of South America, where most of the rest of the world has suffered more than 90% loss. The Chesapeake Bay is an example of one such system, where there's less than 1% of the historic population remaining. And this decline has led to the recognition that restoration efforts are needed. Now, previous restoration has focused on maintaining the oyster fishery, while more recent efforts are focusing on restoring the many important benefits of oyster reefs, such as habitat. Now, determining the importance of these reef habitats and the characteristics that make up a productive reef habitat is a crucial next step for successful restoration. And in this context, a productive reef is one with high diversity and abundance of reef-dwelling organisms on it. And this is where my research comes in. My research asks three main questions. First, what organisms live on restored oyster reefs in the bay, and how productive are these reefs? Does water quality and reef structure affect what lives on a restored reef and their productivity? And does reef structure affect their potential benefit to other fisheries? To answer the first two questions, we conducted a field survey of previously restored oyster reefs in 4 rivers in the Chesapeake Bay. These rivers were the Great Wicomico, Piankatank, Lafayette, and Lynnhaven rivers. In each of the 4 rivers, we sampled 4 restored reefs by embedding benthic settling trays, as shown here, into the reefs. The trays were left for 7 weeks, after which they were retrieved and the samples were brought back to the lab where they were sorted through to remove, identify, and then biomass all organisms. The oyster material in each sample was then sorted into different categories, with oyster clumps, live, unclumped oysters, and dead shell. And these categories are then volumed to provide additional measurements of the structure of the reef. So what did we find? Here are some images of some of the organisms that we found, so you can get an idea of the wide variety of organisms which live on these reefs. In all, 47 different species were found to inhabit these reefs. However, there were differences between the rivers in the species diversity and composition of the organisms that were found. And these differences were found to be largely driven by differences in salinity. We can see in the graph on the left that with salinity on the y-axis and river on the x, that salinity increases as we moved from the Great Wicomico southward towards the Lynnhaven River. And the graph on the right here shows diversity on the y-axis and river again on the x. And you can see that the diversity of organisms was significantly higher in the Lafayette and Lynnhaven rivers, which have the higher salinities, compared to the lower salinity Great Wicomico and Piankatank rivers. Now, how does three-dimensional structure of the reef affect diversity and abundance? To answer this, we determined the relationship between organism density, which is shown here on the y-axis, and some of the various measures of structural complexity that we took. One of the strongest of those relationships was between organism density and the total volume of oyster material shown on the x-axis in this graph. And we can see that as the amount of oyster material increases, so does the density of organisms found in those areas. So based on these two results, it appears that salinity and the amount of oyster material present are two important factors determining the abundance and diversity of organisms on restored reefs. So we saw that organism density increases with increasing structure. However, what does this mean for the big fish that we like to eat? Are they able to take advantage of the increased prey abundance? And to answer this question, I am currently conducting a laboratory predator-prey tank experiment where we are using striped bass as a predator and grass shrimp as their prey. There are 9 treatments resulting from crossing 3 levels of oyster reef complexity, high complexity, which is shown in the top image, and low complexity in the bottom image, and a bare, or a no complexity, which is bare bottom tank, which is not shown here. Crossing those levels of complexity with 3 levels of prey densities, a low, middle, and high. And for each trial, we have 1 striped bass, placing it into each of the treatment tanks for 24 hours, after which it is removed and the remaining number of shrimp are counted to determine % survival. So ultimately, the results from this research will help us to determine the importance of oyster reef habitats in the bay and identify the environmental and structural characteristics that make a productive reef habitat to help us improve our restoration success. Thank you.